The overall goal of the following experiment is to isolate a cell population that is enriched for intestinal stem cells from the midguts of young and old Drosophila using fluorescence-activated cell sorting. This is achieved by first dissecting the midguts of flies in which the cells of interest express GFP. In the second step, the midguts are dissociated into single-cell suspensions, and then sorted by FACS based on differences in their GFP intensity. Ultimately, this gating strategy allows the enrichment for and isolation of intestinal stem cell populations from young and old Drosophila midguts.
Being able to isolate intestinal stem cells from young and from old midguts of adult Drosophila will facilitate the identification of novel molecules in these cells that function during aging. The implications of this technique ultimately extend towards the development of molecular therapies aiming to improve regeneration and organ maintenance during aging. The dissection and tissue dissociation procedure will be demonstrated by Helen Tauc, a graduate student from my laboratory, and the FACS sorting will be demonstrated by Alpaslan Tasdogan, a graduate student from the Institute of Immunologie.
Before beginning the dissection, anesthetize 1 vial of 40 flies with carbon dioxide on a standard fly bed. Then use a razor blade to decapitate all of the flies and transfer them to a dissection dish coated with agarose gel. Pour enough cold PBS plus BSA into the dish to cover the agarose. The flies will float. Then,